Hi guys! Today we're going to talk about the development of the male gametophyte, which when we talk about angiosperms, um, for all practical purposes, we're talking about the pollen. And we'll talk about why I have to qualify that at the end of the lecture. But this first slide is a really beautiful slide showing really the diversity of all of the pollen. And some of these are actually not angiosperms. If you look at this one right here, you can see that there are flagella on it. And so some sperm, not in angiosperms or gymnosperms, are flagellated. Actually, if you Google a fern sperm, they have many flagella. It's pretty impressive. On your syllabus, we're supposed to have a quiz today, but there is no quiz posted for next week, and so I'm going to move our quiz to an in-class quiz next week, and that will cover the alternation of generations and the male gametophyte material that we're going over today. So last lecture we talked about general concepts of organisms that undergo an alternation of generations. And we said that any organism that undergoes an alternation of generations has multicellular sporophyte bodies, which have cells that are diploid, and they also have multicellular gametophyte bodies that have cells that are haploid. And so actually most of this course we're going to spend talking about those gametophyte generations within angiosperms. Now, in some of the plants, the gametophyte generation is dominant. So if you think of mosses like sphagnum moss or liverworts, the green leafy plant body that you see is actually the haploid body. But in gymnosperms and angiosperms, the gametophyte generation has become completely dependent upon the sporophyte generation for protection and nutrition, and it's also become highly reduced. And in the case of the male gametophyte, it is only 2 to 3 cells large. For the female gametophyte in angiosperms, depending on the species, it can be anywhere between 6 and 13 cells. So we've gone from having dominant gametophyte bodies to having microscopic gametophyte bodies. All right, the male gametophyte generation is contained within the anther, within the flower. The female gametophyte generation is contained within the ovule, within the ovary, within the flower. So you can see that these structures or these bodies are actually contained within the sporophyte body. They are wrapped around by the tissues of the sporophyte. So they're being protected and they have nourishment from the sporophyte body. Now, in the case of the pollen, that only lasts until it's mature, because once the pollen is mature, the anthers will open and the pollen is released into the environment so that it can find, hopefully, another plant to pollinate. So the pollen, we'll see, has some adaptations that allow it to survive outside the sporophyte body. So the gametophyte generation in any organism is responsible for producing the gametes, the sperm and the egg cells. And like we talked about last week, in heterosperous species, you have two different types of two different types of spores. In this case, we have microspores and megaspores. And the microspores and megaspores develop into two distinct gametophyte bodies. And so each gametophyte body is going to produce its own type of gamete. The microgametophyte will produce the sperm, and the megagametophyte will produce the egg. And then once fertilization happens, the sperm and the egg come together in syngamy or fertilization, and they result in a new diploid zygote, which then develops into the embryo, which then develops into the seedling and the new plant body, the new sporophyte plant body. So here's an overview of um, the development of the male gametophyte. And the first thing that happens is called microsporogenesis. Microsporogenesis is the formation of microspores. After you have microspores, those microspores germinate into the male gametophyte, and that is called microgametogenesis. So there's two phases in the development of mature male gametophytes. Okay, the first one is meiosis, and that's the formation of spores, taking a 2N diploid cell and producing haploid microspores. That's called microsporogenesis. Then, once you have microspores, which are haploid, you have to develop the mature male gametophyte, and that's microgametogenesis. And the microgametogenesis is considered complete once sperm has formed. And we'll see that in some cases, the mature male gametophyte is the same thing as mature pollen. And that happens when the sperm are formed before the anthers open up. And that's what's known as three-celled pollen. But many times in many plants, the mature male gametophyte doesn't finish developing until after pollination. And so in that case, mature pollen, which is what leaves the plant, is actually still an immature male gametophyte body because it doesn't yet have sperm. It only has the generative cell. And after pollination, that generative cell will split into two sperm cells. And so the mature male gametophyte in that case doesn't develop until it's already in contact with the female plant. All right, so the structure where the male gametophyte develops is known as the microsporangia. And the microsporangia and angiosperms are located inside of the anther. So here is a cross section of an anther, and you can see that inside each inside each stamen, there are four microsporangia. Okay, so this is one anther. It's got two lobes. Each lobe has one microsporangia. Here's another anther. And inside that anther, you also have two lobes with two microsporangia. So the anatomy of the microsporangia consists of the microspore mother cells being located in the innermost tissues. Okay, so the innermost part of each microsporangium will be full of microspore mother cells. These microspore mother cells are the diploid cells that will undergo meiosis. Okay, so these microspore mother cells will only be there before meiosis occurs, so in immature anthers. Okay, the layer of tissue that surrounds those microspore mother cells is known as the tapetum. Okay, if you look at this anther, you can see the tapetum here. And the tapetum is a very important layer of tissue. Those cells provide nourishment to the developing microspores. They produce lots of different enzymes, and they also produce some of the cell wall components that are going to make the outer cell wall of the pollen. Now, the tapetum, because it is a nutritive tissue, you can see here where in this microsporangia, meiosis is already beginning. And so the tapetum has actually separated from the other layers of the sporangium, and it's beginning to be consumed by these microsporocytes. So you can judge how far along in microsporogenesis or microgametogenesis a sporangium is based on the anatomy of the tapetum, based on how disintegrated it is. The tapetum cells are unusual in that they're usually multinucleate cells. They have multiple nuclei, and that allows them to make many, many products very quickly. Okay, so we have the microspore mother cells, or the microsporocytes, in the center. Those are surrounded by the tapetum. The next layer out consists of one to maybe three or four, usually not more than 4 layers of cells. Those are known as the middle layers. The outermost layer is known as the epidermis. Okay, so that's what's in contact with the air. And right underneath the epidermis is the endothecum. Okay, the endothecum. And the endothecum has some functions in opening up the anthers. So here you can see these anthers are opening up, and the endothecum, the cells there will contract in order to rip apart the epidermal cells. So starting from the outside, you have the epidermis as the outermost layer. Directly under the epidermis, you have the endothecum. Between the endothecum and the tapetum, you have the middle layers. There may only be one middle layer, or there may be several middle layers. The tapetum is the innermost wall of the microsporangium before you get to the cells that are going to become the microspores. So the microsporocytes, or the microspore mother cells, are the innermost cells, and they're surrounded by the tapetum, which is that nourishing layer of cells that also produces both enzymes and some of the structural molecules that are required to produce the pollen cell wall. Okay, so here are two different views of the anthers. Here you can see that the tapetum cells are still very much intact. They're still connected to the middle layers. So what stage of development do you think these microsporocytes are in? They're probably pretty young. They probably have either not undergone meiosis yet, or are only in the beginning stages of meiosis. Okay? And if you look at the anthers, this is actually probably more open than actually these anthers are. This flower, at this stage of development, at this stage of the development, the flower bud is still closed and developing. Once microsporogenesis and microgametogenesis are complete, then the anthers open up. In the case of the lily, if you look here, gosh, I wish you could see my hands. The anthers are closed here, and there are these slits down the sides. See these longitudinal slits? That opens up. This slit is going to open up, and actually this anther turns itself inside out. So what is on the inside here is now on the outside here. Okay, so they fold themselves back. And you can see in this image, you can see how the epidermis and all of the other layers, the endothecum and the middle layers, have completely opened up to allow the pollen to leave. Here we have mature pollen, and so this has gone through microsporogenesis and also microgametogenesis. All right, so let's look at microsporogenesis in a little bit more detail. So the cells that begin microsporogenesis are microsporocytes. Those are diploid cells. Okay, you can also call microsporocytes microspore mother cells or pollen mother cells. Those are all synonyms. But you need to remember that these microsporocytes are diploid cells. They're produced by the sporophyte body in the sporangium. And these microsporocytes are the cells that will undergo meiosis to become spores. All right? So meiosis is going to occur. First you get meiosis 1, and meiosis 1 results in what's known as a dyad. These are two cells that are now haploid, but they have twice as much DNA, right? Because we haven't gone through meiosis 2. And they're actually stuck together by a callose cell wall. So these cells are held together even after cytokinesis is complete. After meiosis 1, you get meiosis 2. And so here you can see meiosis 2 has occurred, and now you have what's known as a tetrad. These are 4 haploid cells that have only a single set of chromosomes. Okay? They don't have duplicate DNA like they do in the dyad. At this point, in the point where the cells are in a tetrad, they are still being held together by a callose cell wall. In order for microsporogenesis to be complete, this callose cell wall will be dissolved and each of the individual microspores will be released. Let's look at that in another view. Okay, so here you can see inside this lily anther, you have microsporocytes that are diploid. So you can see some of those here with a single nucleus and no cell plate between them. Meiosis 1 occurs and you get dyads. So you can also see in this lily anther some of these dyads. Here you have 2 nuclei and a cell plate, 2 nuclei and a cell plate. I also want you to look at the tapetum. This is early meiosis. You've had 1 round of meiosis in some cells. The tapetum is still connected to the middle layers in some places. Okay, but in other places it's starting to pull away, and the cells aren't as regular as you see in many plant cells. Okay, they're starting to collapse because their nutrients are being sucked away. Then in meiosis 2, your dyads split into tetrads. Okay, so here you see some dyads, but some of these cells have already divided by meiosis 2, and now you see the tetrads. And notice that the tetrads are still stuck together at this point. Okay, so these are not mature microspores. They haven't been released from the callose cell wall. Now, the tapetum here is in much worse shape. Okay, some of the nuclei are floating free. The cells are ripping away from the middle layers. And in many cases, you can't even distinguish good-looking cells. The cells are collapsing. It's actually that tapetum that's going to be responsible for breaking apart these tetrads. The tapetum is going to release the enzyme callase. Okay, so there's a special polymer called callose, which is the cell wall of these microspores. Callose is similar to cellulose, but it's held together, the glucose monomers are held together by a different type of bond, a 1,3 glycosidic bond instead of a 1,4 glycosidic bond. And so it's different from cellulose. And so the tapetum cells can release callase, and it'll dissolve only the cell wall around these microspores and not the cell walls of the other cells that will have cellulose. So once the callose cell wall has been dissolved around these microspores, then the microspores are singular, and they're going to go on to complete microgametogenesis. Okay, so the process of microsporogenesis is complete once you have microspores, singular microspores that have been released from the tetrad. All right, microgametogenesis is the next phase in the formation of the male gametophyte. Microgametogenesis begins with haploid spores, haploid microspores, and it's complete when you have sperm. So the first thing that happens in microsporogenesis is the microspore grows. It increases in volume, it produces nutrients and other molecules that are going to be necessary for mitosis and the production of the nutritive molecules that are going to be necessary for the pollen to have. A large vacuole is produced. Oh, I'm sorry. And the nucleus of the microspore is pushed by the vacuole to the periphery of the cell. So you can see this is the microspore. It's going to grow. There's going to be a big vacuole, and the nucleus starts to move to the edge. It gets pushed against the edge. And after it actually connects to the plasma membrane, pollen mitosis 1 occurs. So the first mitosis. So instead of one nucleus, now we have two nuclei. Okay. Now this first pollen mitosis 1, is what's called an asymmetric cell division. And it's known as asymmetric because one of the resulting cells gets much, much, much more cytoplasm than the other one. Okay, so here's another scheme. You can see the microspore. It grows. A vacuole, you can kind of see this vacuole here. There's a single vacuole, and that's going to push the nucleus towards the periphery of the cell. Here you can see that the nucleus is actually connected to the plasma membrane, and mitosis is occurring. Okay, once mitosis is complete, you have two cells. One of them is at the periphery of the cell wall here. The other nucleus is within this much larger cell. So here's one cell, much larger cell with this nucleus. Here is the second cell at the periphery. Okay, the cell wall hasn't changed, but now you have two cells. One is stuck to the periphery and one is against that. And I also want you to notice that this large vacuole after pollen mitosis 1 is going to separate into many small vacuoles. Okay, so if you look at, it's kind of hard to see here, but there are lots of little tiny vacuoles now. Okay. All right. After pollen mitosis 1, and you have these two asymmetric cells, one of the cells is stuck to the periphery. This little cell here is going to be engulfed by the larger cell. And this process is similar to phagocytosis. Okay, so this cell here is going to be engulfed by the larger cell, and the larger cell is going to become what we call the generative cell. And this little cell is, the large cell is called the vegetative cell. Sorry. The large cell is the vegetative cell, and the little cell is the generative cell. Okay? The little cell is what's going to split to become the sperm, and the large cell is what's going to germinate to become the pollen tube. Okay? Oftentimes, especially if pollen mitosis 2 occurs after pollination, this generative cell here will take on this very characteristic spindle shape. Okay, it looks like an American football. That's very characteristic of a generative cell, especially those cells that have bicellular pollen. All right, the vegetative cell is that larger cell that split. It contains almost all of the cytoplasm. This here is the vegetative cell's nucleus. Okay, don't mix this up with another cell. Many people, because I will ask you to label a pollen grain. And so you have the vegetative cell and the generative cell and the pollen cell wall. You could label this as the vegetative cell nucleus. But don't label this as the vegetative cell because this is the nucleus. This whole thing is the vegetative cell. Okay, that's a very common mistake. So this vegetative cell will become the mature pollen grain. And once pollen mitosis 1 has occurred, the vegetative cell becomes arrested in the G1 phase of the cell cycle. It is no longer destined to divide. But it does have to produce many, many molecules. It has to produce the nourishment that's going to be required by the pollen grain in order to make a pollen tube. It's going to um, have to make molecules that allow the pollen tube to move through the style. It's going to have to make the molecules that are needed by the generative cell to undergo mitosis to make the sperm. Okay, so here's a list of those. So the carbohydrates and lipids are energy or nutrition. It needs to make any of the transcripts and proteins that are required for the pollen tube growth. And it needs to make osmoprotectants. Osmoprotectants are molecules that protect the cell from drying out. Because remember, the pollen grains have to move outside of the anther. They have to move through the environment, either in the wind or on an insect or some other animal. And so they're vulnerable. And so these osmoprotectants help keep the membranes from drying out. All right. Now, pollen mitosis 2 may happen inside of the anther if you're going to have tricellular pollen, or it's going to happen if you have bicellular pollen. It will happen either on the stigma or within the pollen tube in the style. The generative cell is completely contained within the vegetative cell, and it upon that first mitosis, the chromatin of the generative cell never diffuses. Okay, the generative cell, that's one of the reasons it stays so compact, is that it keeps itself ready for mitosis. It's protecting its DNA so that no damage will arise and affect the gametes. At some point, whether it's inside the anther or whether it's on the female plant, pollen mitosis 2 will occur. And that will split the generative cell into 2 sperm cells. Okay, so after pollen mitosis 2 occur, you no longer have a generative cell because that has split and you now have 2 sperm. Now it's really interesting because the generative cell maintains a cytoplasmic connection with the vegetative cell's nucleus. Okay, first there is a connection between the cytoplasms, and then later on in the development of the pollen grain, that cytoplasmic connection joins to the nucleus of the vegetative cell. And that's important because once the pollen tube starts to grow, it's the vegetative nucleus or the pollen tube nucleus at that point that joins up and stays as a single unit with those sperm cells. And these 2 sperm cells plus the vegetative nucleus, which are all connected to each other by this cytoplasmic connection, is known as the male germ unit. Do I have that? I thought I had that written down somewhere. Anyways. All right, so once pollen mitosis 2 has occurred, you have a mature male gametophyte. Okay, the mature male gametophyte is only present once the sperm have formed. Okay, so in some cases, in many cases actually, the mature pollen is not the mature male gametophyte because you don't have sperm. If you have bicellular pollen, you don't have sperm. It's not a mature male gametophyte. Okay. About 40% of angiosperms shed tricellular pollen. And tricellular pollen is when the pollen mitosis 2 occurs inside the anthers and you have a pollen grain that has sperm already leaving the anther. All right, so let's back up. We've talked about the formation of the pollen in terms of the nuclei and the cells inside, but a very important part of the pollen formation is the pollen cell wall. And actually, when you look at pollen, this is what you see. Um, the synthesis of the pollen cell wall begins before microgametogenesis begins. It begins shortly after meiosis II is complete. Okay, so before the callus cell wall, um, um, so while the pollen grains are still in the tetrad, um, there's two layers to the pollen cell wall. The exine is the outermost layer, and it's composed of this really amazing molecule called sporopollenin. Sporopollenin is one of the hardest substances on Earth. There are no enzymes that are known that can degrade it, and in fact, we have ancient pollen that has been recovered from Antarctica and from other ice core samples, and all that's left is the sporopollenin. The inner layer of the pollen grain is called the intine. The intine composes a complete layer around the pollen cell wall. We'll see in a minute that the exine is incomplete because it has to have these germination pores so that the pollen tube can grow. The intine is composed of cellulose and pectins, so it's much more similar to the to a normal cell wall. And then because sporopollenin is so tough, all pollen grains have what are called apertures or germ pores. Okay, apertures is a synonym for germ pore, and those are these holes within the exine that allow the germination of the pollen tube. All right, this is actually fairly recently discovered in Sweden. This is the oldest fossil of an angiosperm, or at least something that is angiosperm-like. This pollen so these are the same pollen. This is when it was first pulled up, and then they hydrated it, and this is what it hydrated to look like. This was dated to 240 million years ago. That's the early Triassic period, and this is actually way earlier than they first thought that angiosperms had evolved. So this was quite a discovery by the University of Zurich. The only thing that's left here is the sporopollenin, and it lasted 240 million years. That is just amazing. All right, the exine produces all these cool ornamentations. When you look at micrographs of pollen, they're quite beautiful. But unfortunately, those ornamentations are also responsible for giving you allergies. So hay fever, when certain plants are spewing their pollen into the air, it goes up your nose, and those ornamentations are highly irritating. Nobody's really sure exactly how the ornamentation is determined, but they think it has to do with the original framework of the callose cell wall in the microspores. The deposition of sporopollenin begins before the microspores are released from the tetrad. And then once they are single, once the microspores are released from the tetrad and they're single cells, then the cell wall production increases rapidly. And in fact, it has to wait somewhat until the microspore has increased its volume before pollen mitosis 1. So once the once the microspore has achieved its final volume, that's when sporopollenin can really be deposited in quantities and complete its deposition. It is the tapetum that is responsible for the synthesis and deposition of sporopollenin. And this is the reason why I always say the pollen grain is more or less the male gametophyte because the sporopollenin is not made from haploid cells. Remember, the tapetum is part of the microsporangium. The microsporangium is part of the sporophyte body. The tapetum is full of diploid cells. The tapetum is composed of diploid cells. And because of this, the sporopollenin comes from the sporophyte body, not from the gametophyte body. But this is really the only component of the pollen grain that is not part of the gametophyte body. All right, it's an important part, but it's not even the complete cell wall. It's only one part of the cell wall, okay, the sporopollenin. All right, because exine is such a tough material and it cannot be degraded by enzymes, there has to be holes in the exine that allow pollen tubes to grow. And those holes in the exine, or the outer layer of the cell wall, are known as apertures or germ pores. Okay, apertures and germ pores are both good words for these holes in the exine. Oftentimes, the apertures are laid down at the meiotic spindle poles. So um, where the spindles would arise from during meiosis of the microsporocytes, that's where the germ pores end up. And this is one of the reasons why it's also believed that it's the microspore that kind of dictates um, how the ornamentations of the exine are determined. The intine, the inner layer of the pollen cell wall is the intine, and the intine begins its deposition at the apertures. And then after it's deposited at the aperture, it begins to spread and cover the entire microspore. All right, so the pollen cell wall consists of two layers: the intine, which is the inner layer, which is composed of cellulose, hemicellulose, which is pectin, and callose, and the outer layer, which is the very, very hard exine, composed of sporopollenin. All right, finally, anthesis. How do these pollen grains, the male gametophytes, get released into the environment? And that's known as anthesis, the opening up of the anthers, or the opening up of the sporangia. So when the pollen is mature, the anthers will open, and different species open in different ways. These are, so we already saw the lilies that open via slits, and actually after the slit opens, it folds itself inside out. These pentas open up also by longitudinal slits, but they don't fold. They just open up, and here you can see some of the pollen grains. These eggplants are opening by by pores. So you have these little pores at the ends of the anthers, and the pollen comes out of those little pores. All right? Even in related species, you can have different mechanisms of anthesis. So these are two different eucalyptus, and they're in two different clades, but they're all eucalyptus. They're in the same genus. These eucalyptus here have these terminal slits rather than the longitudinal slits we saw earlier. These eucalyptus, on the other hand, they have pores, and the pollen is just kind of squirting out of those pores. That's pretty cool, huh? All right, this is the last slide, yes. So, so there's no quiz today. I would like you um, for just for part of your um, assignment 2, I would like you to look up 3 different species of pollen and draw what they look like. Okay, it doesn't have to be beautiful. They can be just ovals with slits or circles with spikes. But draw 3 different species of pollen. And I want you to indicate where are the apertures. Okay, and what do they look like? Are the so also the apertures, so here you can see here's an aperture that's a slit, and then some apertures have pores like this one. Okay, and make sure you at least somewhat, I want to see what the ornamentation looks like. Okay, so try to get some ornamentation on there. I'll give you a hint that if you're a really bad artist, there are some pollen that have very little ornamentation. um, all right, so next week I will be back in class. I'll be very jet lagged, but I'll be there, and we're going to have a quiz. We will not have our first presentation at that point. We're going to push off that first presentation so that I have a chance to meet with the group. And then we will have our lecture on bicellular and tricellular pollen, and not bicellular, tricellular, I'm sorry, different pollination issues, okay, issues surrounding pollination. So, all right, I will see you next week, and I hope you have a good evening.